Youth Biolab Jeunesse.
Hi, I'm Steve.
I'm Megan.
And I'm Anjelica.
We're from the Youth Biolab at the Albrechtsen Research Centre at St. Boniface Hospital. We all know the job of the hospital is to treat patients. Here at the Research Centre, our job is to study disease. If we ever want to cure a disease, we have to understand it first.
When you think of scientists studying disease, you probably think first of things like cancer or more recently, COVID-19. We don't study either of those here. But there are research scientists over at the National Microbiology Lab who are studying infectious diseases like the flu or COVID-19 or Ebola, where they have special equipment that can protect them from the germs that they're working with. There are also groups of cancer scientists working over at Cancer Care Manitoba in Health Sciences Centre, and it's their job to study cancer. We like to keep scientists together so that they can work as a team to come up with cures or treatments for these diseases that we see.
At the Albrechtsen Research Centre, we have three big interests. On our fourth floor, we have the Division of Neurodegenerative Disorders. This is where we study diseases that affect the brain and nervous system, just like Alzheimer's disease and type 2 diabetes nerve damage. On our third floor, we have the Institute of Cardiovascular Sciences. This is where we study diseases that affect the heart and cardiovascular system. On our second floor, we have the Canadian Centre for Agri-Food Research in Health and Medicine, and this is where we study food. They research foods that are really good for us, some that might be bad for us, and some that might be so good that they act like medicine.
Our lab, the Youth Biolab, was created so that we can share some of the things that we do here in the Research Centre with you. And hopefully we can help you learn a little bit more about how medical science works and understand a little bit more of the science of your own health.
Hi everyone. Today we're going to talk about microscopes. When scientists are studying disease, they need to use special instruments to understand what's going on inside of the body. Throughout history, many people have developed different types of instruments to help us see things that are too small or too far away. You've probably used at least one of these in your life. We use magnifying glasses to make tiny little bugs look much bigger. If we want to see a bird that's really far away, we can use binoculars to see them, and they look much bigger and closer. And if you want to look at a star that's so far away it looks like the size of a pea, you use telescopes to make that pea-sized star look big enough to see all of its details. All of these tools are examples of optical devices which use lenses to bend light in different ways to make things look bigger or closer. In the lab, we use tools that kind of do the same thing. One of the coolest optical devices that we get to use in our lab is a microscope. The name is quite fitting because microscope means extremely small, and we use microscopes to see extremely small things. The lenses in microscopes make small things look bigger. Let's take this lens for example. If you look at it on its side, it's thicker in the middle than it is around the edges, so it bulges out in the middle on both sides. This is called a convex lens. Convex lenses bend light in a special way to make objects look larger and closer. So if we want to make a cell look larger and closer, we need to use a convex lens. So if I put this lens up to my eye, it makes my eye look bigger than it normally does because of the way this lens bends light. Microscopes usually have more than one lens to magnify things. So if I add another lens, my eye looks way bigger than it normally does. At least I hope that's not what my eye looks like. There are other types of lenses that are thinner in the middle and thicker around the edges, so it indents in the middle on both sides. This is called a concave lens. These lenses bend light to make objects look smaller and farther away, which is definitely not good if you want to look at really small things. Here is a slide of a mosquito head. It is super small and hard to see with our bare eye. This mosquito head is sandwiched between two pieces of glass, one on the top and one on the bottom. We've all seen a mosquito and we know how small they are, but maybe you've seen one close up. You could probably see their eyes, maybe their sucker, but you couldn't see much else. With a microscope, we will be able to see what their head looks like closer in detail. we're going to use a compound light microscope today. You probably use these in your school, and it has all the basic parts every microscope has. So we have our slide, and the first thing we're going to do is place it on the stage, which is just a platform that holds our slide. I'm going to place the mosquito head over the hole inside of the stage. If I look into the microscope, I can't see anything. It is way too dark. We have to turn on the light. We have a light source at the bottom, and the light shines through the hole in the stage, through the slide, up through these parts, to the eyepiece. This is an eyepiece or ocular lens. This is what we look through to see our slide. The lens inside of here is 10 times magnification. So what that means is whatever we're seeing through this lens makes things 10 times bigger than what we would see with our bare eye. We also see through another set of lenses as we look down on our mosquito. These are called our objectives. Each objective has a different magnification lens inside. This red one is a 4 times magnification, the yellow is a 10 times magnification, and the blue one is 40 times magnification. When we use the 4 times objective and look into the microscope, we're seeing an object that is made 4 times bigger by this objective, and then another 10 times bigger by our ocular lens. So the image that we're seeing is 40 times bigger than the original thing. Just like when we have one lens on my eye looks big. But when we use 2 lenses, my eye looks way bigger. When we look on our slide, our image is a little blurry, so we have to focus it with our coarse focus knob on the side. And this just moves our stage up or down in like large increments. We only want to use the coarse focus when we're on the smallest objective, because this movement is way too much if we're zoomed in on our 10 times objective or our 40 times objective. As I turn the knob, we can see that I'm bringing the mosquito head into focus, and we can see it clearly now. So now that we see it clearly, we can see our sucker in the middle. We can see there are 2 antennae with hairs on either side. And we can also see that they have really big compound eyes. And these eyes are made up of thousands of little tiny lenses. We definitely wouldn't be able to see this in such close detail without a microscope. If we zoom into 100 times magnification, we might have to use the smaller fine focus knob that only moves the stage a little bit at a time to help us focus on our slide. The mosquito head is actually too thick to focus on the whole thing at once, but as we move the fine focus knob, you can see that we're focusing on different layers of all of the lenses on their eye. Now let's take a look at something even smaller than a mosquito head. We're made up of tiny living things called cells, and we can collect some to see what they look like under the microscope. There are a few places we can easily get a sample of live cells. I'm going to take this toothpick, gently scrape some cells from the inside of my cheek, and put it on this slide. I have this cover slip, which is just a piece of glass that I'll put on top of my cells to keep them flat and easier to see. Now we have some cells on this slide, but it doesn't really look like there's anything on there. The slide looks clear, and you can't see the cells at all with your bare eye because they are so small. Let's take a look at them underneath the microscope. We can see that in my microscope, my cheek cells look nice and round and plump, but they're a little hard to see because they're so clear. And that's why we couldn't see them on the slide when I made it. Scientists have solved this problem by using stains. Have you ever tie-dyed a shirt? You have a bunch of colors to choose from to turn your plain white shirt into a cool, colorful one. Scientists do the same thing. We can turn our clear cells different colors. When cells are colored, we can see them a lot better. So if I make a new slide with my cheek cells, but stain them with the blue dye, we'll be able to see the cells way better. Wow, that is much better. We can see my cheek cells in more detail. You can see the nucleus and you can see the cell edges are much more defined. Cells don't always have to be stained blue. There are different colors we can choose from to help us see our cells better, like pink or even red, for example. Scientists can use special types of stains that only dye specific structures of the cell that the scientists want to study. Stains are awesome, but there's one little problem. If we want to keep cells alive, we can't stain them. Stains are super toxic to cells, so to get around that problem, we have to use different types of microscopes to see living cells without stain. When we look at living, unstained cells, we don't see too much in a light microscope. That's because the clear cells don't absorb a lot of light. So all of the light coming from the microscope is just shining right through the cells. We have two phase contrast microscopes over here, and they still have light and lenses, just like a compound light microscope, but they also have these special rings inside. And these help give contrast to clear cells. The rings inside move light in a special way to help us see the detail on the surfaces of cells and see the cell's contents that we could not see with our light microscope. Thanks to phase contrast microscopy, scientists are able to see the living cells without staining them. If I take my unstained cheek cells and put them under a phase contrast microscope, we can see the same amount of detail we saw earlier when we turn these cheek cells blue, except in this case, we don't have to stain them at all to see them in detail. Here we have our fluorescence microscope. If labs want to look at the things that are going on inside of the cells, they can use special glowing tags to attach to specific cell parts. Scientists can tag parts of the cell that they want to study with a special molecule called an antibody, and this antibody glows when you shine a special light on them. To see the tag, we use a microscope called a fluorescence microscope. And these microscopes have light and lenses just like a light microscope, but they use a different type of light that we can't see, like UV light. When we use different light and different antibody tags, we can make different parts of the cell glow in different fluorescent colors. They make really pretty images, and scientists use this glowing light to help them see how diseased cells change over time. The next microscope we want to talk about today is the confocal microscope. These microscopes still see fluorescent antibody tags, but they use a different kind of light. Confocal microscopes use thin lasers instead of light. These lasers focus on very small parts of the cell at a time. A mirror moves the laser to point and scan throughout the cell, taking tiny but very detailed pictures of each section. If this Rubik's Cube was the cell on my slide, the mirrors would move the laser here and take a picture, and then the laser would move here and take another picture, and it would keep moving to all the different pieces of the Rubik's Cube. Once the microscope has captured all of the image of the cube at this layer, it moves on to the second layer of cubes and takes pictures of each of those 9 cubes, and then moves on and takes pictures of the cubes at the bottom. Now, when the computer program stacks all of the pictures together, we get a 3D image, just like this Rubik's Cube.
Hi, I'm Vicky. I'm working in Lori Kitchenbaum lab. I'm taking confocal pictures for cancer cells right now. And we study difference between cancer cells and cardiac cells in cell culture.
The last microscopes we want to talk to you about today are the scanning electron microscope, or SEM, and transmission electron microscope, or TEM. Remember how I said all microscopes use lenses and light? Well, these microscopes are a little different. They use an electron gun to shoot electrons. Electrons are very tiny particles, and we use those instead of a light source. In SEM, electrons are shot on top of your sample, and as they hit the sample, they bounce off at different angles. An electron detector measures all of those electrons that have bounced off and gives you an image that looks like this. A nice high-resolution 3D image. SEMs allow researchers to take high-resolution pictures to see the outside of very small things. TEMs are a little different. They allow researchers to take high-resolution pictures to see the inside of small things. Scientists need to take a super thin slice of a cell for electrons to pass right through. The transmission electron microscope has a little sensor underneath this super thin section of your cell and analyzes the amount of electrons coming through your sample. SEMs and TEMs are not only special because they use electrons instead of light, but they can magnify an image to a much higher magnitude than any light microscope can. SEMs can magnify an image up to 2 million times its size, and TEMs can magnify an image up to 50 million times its size. So these microscopes make your image look way bigger and way nicer, and this helps scientists learn more about the things we can't see with our bare eye. Thanks for tuning in to Healthy Curiosity. Stay curious. Make sure to click like and subscribe for more videos and follow us on Instagram and Twitter. To support the Youth Biolab's ongoing programming and expansion plans, please contact St. Boniface Hospital Foundation at stbhf.ca.